Welcome to this presentation. I'm the President and Primary Biochemist of Moondust Cosmetics Limited. This story really starts at the beginning of the coronavirus pandemic in 2020, with the Public Health Agency of Canada sending out letters to science tech companies asking them to contribute information about any of their products that may be useful in combating COVID-19. So at first I was a little surprised by this request because we specialize in making sunscreen. But then I started researching SARS-CoV-2, and what I learned was really quite exciting. So just to give you a little background, zinc oxide is the broadest spectrum UVA and UVB reflector that is approved for use as a sunscreen by the FDA. It acts as a physical sunblock by scattering ultraviolet light more effectively than other substances, and unlike many other sunscreens, it effectively prevents apoptotic sunburn involving UV-induced cell death, thereby helping to protect against skin cancer. Most sunscreens only prevent tanning. While I was doing some research on depleted uranium and uranium detoxification for a paper, I realized that zinc is absorbed from our topically applied sunscreen quite effectively, and may be utilized in metallothioneins, which are involved in helping the human body to detoxify such heavy metals as uranium. In the case of SARS-CoV-2, an excess of intracellular zinc ions has been found to efficiently inhibit the RNA synthesizing activity of the RNA polymerase replication and transcription multiprotein. Enzymatic studies in vitro have revealed that zinc directly blocks the activity of the polymerase. So there is a small charitable clinic in India which we sponsor, and they wanted to reopen after the mid-June 2020 lockdown last year. So I discussed the possibility of carrying out a preliminary clinical trial with zinc supplementation, although it was not possible to send them our zinc oxide containing sun care product from which the zinc is effectively absorbed due to the restrictions. But they agreed to carry out a preliminary clinical trial with oral zinc supplementation. Just until recently, India has had the second highest COVID-19 infection rate in the world after the US. However, up until now, the mortality rate has been comparatively lower than in the US, possibly due to a relatively younger average age of the general population. This purely observational two-part study to establish dosage and tolerance of the zinc vitamin and mineral supplementation was carried out at a private clinic located in a small town in North India. The clinic that took part in this trial employed a total of 15 staff members, 9 men and 6 women. They included 2 doctors, 1 male, 1 female, 3 nurses, 2 laboratory technicians, 1 security guard, 1 cleaner, and 6 general maintenance staff. All participants consented to take part in the study, although the medical staff, 7, were aware of the potential benefits of supplementation. The non-medical staff members, 8, were not aware of the potential health benefits. However, they were informed that the supplementation was not harmful in any way. Each staff member took part voluntarily in this trial. Part 1 was initiated in July 2020 and extended to December 2020. Part 2 was initiated in January 2021 and extended until March 22, 2021. It is unlikely that the townspeople were taking oral dietary supplements of any kind, since they are not that popular in India, and provided the background population for these studies. At the time of both studies, active coronavirus cases were present in the community or vicinity and formed a basis for comparison. In addition to standard precautionary measures adopted universally during the pandemic, including the use of masks, hand sanitizers, and physical distancing, a careful selection of vitamin and mineral supplements was made to help protect the hospital staff from coronavirus infection. The supplements selected for the staff included a combined daily dose of vitamin C and zinc, and a weekly dose of vitamin D capsules. The reason for this choice of combined supplements at above biological doses was as follows. Vitamin D enhances cellular innate immunity partly through the induction of antimicrobial peptides, including human cathelicidins and defensins. Cathelicidins exhibit direct antimicrobial activity against a spectrum of microbes, including many types of bacteria, enveloped and non-enveloped viruses, and fungi. Moreover, vitamin D is effective in reducing concentrations of pro-inflammatory cytokines that produce the inflammation that injures the lining of the lungs, leading to pneumonia during viral infections like COVID-19, and increasing concentrations of anti-inflammatory cytokines. Vitamin D deficiency is a worldwide problem, but is particularly pronounced in the elderly who are at risk of contracting severe COVID-19 infection. Vitamin D modulates the presence of pro-inflammatory cytokines in the body by preventing macrophages from releasing too many of these. Thus, it is not surprising that vitamin D deficiency has been correlated with COVID-19 cases and an increased risk of mortality in a large European study and in a number of other recent studies. Zinc. RNA synthesis occurs in the life cycle of the SARS-CoV-2 virus in order to reproduce its genetic material, and is catalyzed by an RNA-dependent RNA polymerase, which is the core enzyme of a multi-protein replication transcription complex, and is well conserved among the members of the coronavirus family, including SARS-CoV-2. Enzymatic studies in vitro have revealed that zinc directly blocks the activity of the RNA polymerase. In the human body, zinc displays antiviral effects by modulating the type 1 interferon response and performs a variety of viral antioxidant functions. Inside the cell, the harmful effects of free radicals are balanced by the action of antioxidant enzymes such as copper zinc superoxide dismutase and non-enzymatic antioxidants such as metallothioneins, which utilize zinc and help to regulate its intracellular levels. There are a number of other ways zinc functions in both adaptive and innate immune systems. It regulates the proliferation, differentiation, maturation, and functioning of lymphocytes and other leukocytes. Moreover, zinc deficiency may be present in 17% of the world population. Interestingly, a trial with 4 COVID-19 patients suggested that therapy with high-dose zinc salt oral lozenges up to 200 milligrams per day initiated significant reduction of disease symptoms within 24 hours. Short-term zinc use at these doses is considered safe. Thus, low-risk ways of increasing zinc bioavailability in the body above biological levels can be safely considered. Vitamin C. Vitamin C is known as an essential antioxidant that efficiently quenches damaging free radicals produced during the course of normal metabolic respiration by the body. In addition, the anti-inflammatory action of vitamin C, ascorbic acid, is evidenced in a number of cytoprotective functions under physiological conditions, including the prevention of DNA mutation induced by oxidation. Moreover, vitamin C is well-known antiviral agent that has been demonstrated to show antiviral immune responses, especially against influenza virus at the onset of infection, by the increased production of interferon alpha and beta. There has also been some interesting evidence that oral vitamin C may reduce the incidence and duration of respiratory infections, while intravenous vitamin C has been shown to reduce mortality, ICU and hospital stays, and time on mechanical ventilation in severe respiratory infections. Vitamin C deficiency has been observed in many respiratory infections as well, and a recent study from New Zealand reported that patients with pneumonia had depleted vitamin C levels as compared with healthy controls, suggesting that it may be potentially helpful in the treatment of COVID-19 at therapeutic doses. On average, the clinic was visited by 60 patients per day during the July to December 2020, January-March 22, 2021 trials. Thermal scanning was instituted at the clinic gates, and any patient with a fever was seated outside and given a week's supply of vitamin C plus zinc tablets, vitamin D capsules, and aspirin. The patients without fever were allowed inside the clinic compound after receiving hand sanitizer and a disposable mask. They were instructed to keep a 2-meter distance between themselves and other patients as they waited on chairs outside the clinic. Only 6 patients were allowed into the clinic at one time, 10 to 12 being the usual number. All patients with cold symptoms other than fever also received oral vitamin C, zinc, and vitamin D supplements. All the hospital staff wore medical masks. PPE was not considered necessary as there were relatively few coronavirus cases locally, so the hazard was not deemed to be extreme. Infected individuals in the district were immediately removed to pre-designated quarantine locations by government health inspectors, where they received medical treatment for 2 weeks. There were no adverse reactions to the special selective supplementation regimen in any of the staff members. There were no COVID-19 cases recorded among any of the staff members for the duration of both preliminary studies. even though approximately a third of them were living in and commuting from nearby towns and cities where the incident rates were higher. The length of this two-part trial suggests that there was ample opportunity for COVID-19 infection to occur in any of the subjects, especially since routine social distancing was not being observed during much of this time in India. Similarly, the townspeople often traveled on public transport to other towns and cities and would have been exposed to potentially infected individuals at social gatherings, just like the staff who participated in this study. Coincidentally, no COVID-19 cases occurred among any of the patients who were treated at the clinic during this trial either. A small two-part clinical trial with 15 and 14 subjects respectively was undertaken to test the feasibility of taking specific supplements with antiviral properties to aid in the prevention of COVID-19 infection, vitamin C, D, and zinc. It was concluded that this type of targeted supplementation of medical professionals and healthcare workers in an environment of potentially heightened exposure to coronavirus could be beneficial at the established doses, which were non-biological therapeutic doses. The combination of vitamins and minerals included in this study was selected for its special biochemical qualities in potentially combating the coronavirus. No other supplements aside from these three were provided to the participants during the two-part trial. Even though a variety of vaccines did become available following this two-part study, and all the clinic staff opted to be vaccinated, it was decided to continue with this special supplementation regimen even after the 6-month and 2 3/4 month trial period for several reasons. Firstly, it is not known exactly how long the antibody immunity generated by these vaccines lasts, and therefore there may be a lag period when people are not protected. Secondly, as new variant strains of the novel coronavirus appear, the vaccines may become less effective or even ineffective at some point. Thus, it is preferable to seek protection simultaneously from a number of sources that attack different parts of the viral machinery. Thirdly, even though it is possible to alter existing vaccines to target these new variants, it takes some time to re-engineer them, so it is not desirable for recipients to have an unprotected interval. Finally, there is the question of how many vaccines an individual can safely receive each year without engendering negative physiological consequences or possibly even an antibody overload. Unfortunately, the small sample size, 14 and 15, of the test group in this two-part trial could not be rigorously analyzed statistically in relation to the much larger population in the town, which formed a backdrop. At the same time, it is likely that other hygienic practices adopted at the clinic during the pandemic may have contributed to the positive results. However, it is more than likely that the clinic staff were exposed to infected persons during the study. Therefore, some interesting insights into supplementation with specific vitamins and minerals of medical personnel may have been gained. They may prove useful for further clinical research into COVID-19 prevention, and be particularly relevant for lower income countries without immediate access to vaccines, or in countries like Chile, where vaccines are proving to be less effective despite an extensive vaccination program. Thanks for attending this session. You can follow the link for more information.